Hello everyone, welcome back to the class. In our previous class, we had learnt about mitochondria, which is an organelle that the term of that organelle was coined by C. Benda. It is an energy converting organelle. It is present in all eukaryotic cells. They are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are popularly called as powerhouse of cells. They are membrane bound, double membrane bound in fact, and are plastic organelle. They are having the intermembrane space between outer and the inner membrane. The outer membrane is fairly smooth, which protects the organelle and contains specialized transport proteins such as porins. Mitochondrial porins or voltage dependent anion selective channels allow the passage of small molecules across the mitochondrial outer membrane. The inner membrane is highly convoluted, forming folds called cristae, which greatly increases the membrane surface area. They are very impermeable to many solids due to the presence of high content of phospholipid called cardiolipin. They have two phases, that is towards matrix phase, which is N phase. N is referred as negative, and cytosolic side, that is P side, which is positive. Inner membrane contains enzyme complex called ATP synthase, which is the complex known as oxysome or F0 F1 particle, which makes ATP. The matrix is having the large internal space, and it contains multiple copies of double stranded DNA, which is circular, mitochondrial ribosomes, tRNAs, and mitochondrial proteins. So this is the structure and the labeled diagram of mitochondria. Mitochondria is termed as semi-autonomous organelle. Why? They have their own genetic material referred as mitochondrial DNA, which is mtDNA, and they also possess their own ribosome, which is helpful for the synthesis of their own protein, and the organelle dynamically or frequently dividing, fusing, and changing its shape. The mitochondria multiply by binary fission, which is very similar to prokaryotic character, and it suggests that mitochondria has evolved from bacteria, which is a purple photosynthetic bacteria specifically, about 1.5 into 10 to the power of 9 years ago. So the mitochondria which has been evolved from bacteria, this term or statement has the theory, which is endosymbiotic theory, which was proposed by Lynn Margulis. According to this theory, mitochondria are supposed to have evolved in eukaryotes from endosymbiotic association of purple photosynthetic bacteria. So first, the captured cell or the endosymbiont was then reduced inside the inside this eukaryotic early eukaryotic aerobic cell, where it reduces to a functional organelle bound by two membranes. Evidence to support endosymbiotic theory is one, binary fission, and second, it has a self-replicating body, and it has own DNA, and it has protein synthesis. The protein synthesis in mitochondria is inhibited by variety of antibiotics, which is giving the hint or the similarity between bacteria, and mitochondria are approximately the same size. And mitochondria are inherited cytoplasmically. Majority have the uniparental inheritance, where female gametes mostly contribute mitochondria. In some eukaryotes, both parents contribute mitochondria to zygote. Hydrogenosome, it is a specialized organelle in protist, especially in Trichomonas vaginalis, adapted for anoxic or microaerobic condition in which they lack mitochondria. They have this hydrogenosome. Hydrogenosomes are the site of pyruvate fermentation, which is involved in synthesis of ATP and hydrogen. Pyruvate is broken down to acetate, carbon dioxide and molecular oxygen. These are surrounded by soluble membrane and produce ATP. They produce molecular hydrogen through fermentation. They lack cytochromes and usually lack DNA. We discussed the mitosome also. These are found in organisms that do not have mitochondria. It is termed as highly reduced or cryptic mitochondria. It is present in unicellular eukaryotes, which is entamoeba, histolytica and microsporidia. So till this we had discussed in our previous class. Today we will be discussing about targeting of mitochondrial proteins. Mitochondrial proteins are synthesized by 80S cytosolic as well as 70S matrix ribosome. 99% of mitochondrial protein are encoded by nuclear genes and are synthesized as precursors on cytosolic ribosome. Proteins synthesized by cytosolic ribosomes are translocated into mitochondria post-translationally. Proteins imported into the mitochondria may be located in outer membrane, intermembrane space and inner membrane or matrix. So before getting into the transportation or translocation of protein, let us understand the structure of mitochondria. As we have already discussed this, it is having the outer membrane which is complete and which is smooth, and it is having inner mitochondrial membrane which is having convoluted foldings, that is called cristae, and there is the inside called matrix. This is matrix, this is inner membrane, this is outer membrane. The space between the outer membrane and inner membrane is intermembrane space. Intermembrane space. Whichever proteins which are meant to or designated to mitochondria, it may stay in the outer mitochondrial membrane, or it get inside the inner mitochondrial membrane, or it may be target to the matrix. So there are three possibilities in mitochondria. So the proteins can be targeted to outer mitochondrial membrane or inner mitochondrial membrane or matrix. So there are three possible ways, and all these proteins which are designated to outer, inner, matrix, all these proteins will be having a specific target sequence, and there are some receptors which receives the particular protein and will stop the further translocation so that the protein can get embedded inside the particular membrane. So let us now understand how the translocation of mitochondrial proteins happen. So to begin with, mitochondrial proteins or the proteins which are to be transported to the mitochondria, they are initiated. The importing of mitochondrial proteins is initiated by binding of mitochondrial target. I'll write MTS, mitochondrial targeting sequence, to the import receptor. So this is a specific sequence. Remember, there was a protein and there was a sequence attached to it. That sequence is MTS here. In the case of mitochondrial protein translocation, the protein will be having mitochondrial targeting sequence. This mitochondrial targeting sequence will bind to the import receptor. This import receptor is present in the outer membrane. So this binding of mitochondrial targeting sequence to the import receptor initiates the importing of the mitochondrial protein. So there are two distinct translocation complex. Two distinct translocation complex based on the environment or based on the presence of this translocation complex. So there are two translocation complex, namely Tom complex and Tim complex. Tom complex stands for translocase of outer membrane. Translocase for translocase of outer membrane. It means it is a translocase enzyme complex or translocation complex which is meant for outer membrane. This TIM is translocase of inner membrane. So there are two translocating complex, distinct translocative complex that mediate translocation are situated in the outer and inner mitochondrial membrane. The translocation complex which is present out in the outer mitochondrial membrane is termed as TOM, and the translocation complex which is present in the inner mitochondrial membrane is TIM. Okay. So let us now first discuss about Tom complex. Tom complex is present where? It is present in the outer membrane. They consist 7 different subunits. They are made up of 7 different subunits, and they have these parts. That is receptor. Receptor in the sense it is having Tom 20, Tom 22, Tom 70. So these components will recognize the precursor protein and transfer them to central component channel, forming Tom 40. Okay, and it is also having, this Tom complex is also having protein in them. That is Tom 5, Tom 6 and Tom 7. They are involved in the assembly and dynamics of the Tom complex. So what all this Tom complex contain? It contains 7 subunits and it contains receptor. The receptors are Tom 20, 22, 70. These are the components of the receptor which recognize the precursor protein and transfer them to the central component, the channel forming Tom 40. And also it contains Tom proteins, that is Tom 5, Tom 6, Tom 7, involved in the assembly and dynamics of the Tom. So this was about Tom complex. Let us now learn about Tim complex. Tim, where it is present, Tim complex is the distinct translocation complex present in the inner mitochondrial membrane. So there are two Tim complex, that is Tim 23 and Tim 24. Tim 22. So they transport different classes of proteins. Tim 23 complex is formed majorly by 3 essential inner membrane proteins, IMP, inner membrane proteins. That they are Tim 50, Tim 17, Tim 23. All this inner membrane protein will form a Tim 23 complex. The function of Tim 50 is, Tim 50 inner membrane protein is, it acts as a receptor. It has the receptor function in the intermembrane space. It recognizes the protein and it receives. Then in Tim, the function of Tim 17 is, it acts as a motor recruitment. It helps in drawing the protein inside. And Tim 23 is a channel forming protein. So Tim 50 is a receptor in function which receives. Tim 17 is a motor recruiter, and Tim 23 is channel forming protein. So this was about two different translocation complex which is present in the mitochondrial membrane. So this is just a quick revision. Import is initiated by the binding of mitochondrial targeting sequence to an import receptor in the outer mitochondrial membrane. So there are two distinct translocation complex, TOM and TIM. TOM, they consist 7 different subunits, receptor 20, 22, 70, and TOM proteins, that is 5, 6 and 7. TIM is our two types, TIM 23, 22, transport different classes of proteins. TIM 23 complex is formed by 50, 17, 23. TIM 50 receptor function, 17 motor recruiter, TIM 23 channel forming. So TIM 22 complex contains again 3 inner membrane proteins, that is 18, 22, 54, same as TIM 23 complex. Let us now see the structure of this TIM and TOM. See, this is the TOM. Whichever components I mentioned in the earlier slide, it is present here, that is receptor 20, TOM 20, 22 and 70. These receptors will recognize the precursor protein and transfer them to the central TOM 40. Okay, next TIM. Do we have TIM here? No. In the coming slides we'll be having. So next, now we learn about mitochondrial target sequence. So what is targeting signal sequence? It is a specific sequence which helps to recognize and which helps to designate the proteins to the particular location. So mitochondrial proteins is also having a particular targeting signal sequence. Mitochondrial proteins which translocate from cytosol to mitochondria contains the mitochondrial targeting sequence. It is having MTS, as I discussed before. It is having MTS, mitochondrial signal sequence. This mitochondrial targeting signal sequence are of two types. One is N-terminal cleavable matrix targeting sequence. I'll write it as TS in short. Matrix targeting sequence. It is also known as pre-sequence. SE. So first type of MTS or the mitochondrial targeting signal sequence is N-terminal cleavable matrix targeting sequence, that is pre-sequence. And the another one is non-cleavable internal targeting sequence. So there are how many? Two types of mitochondrial targeting signal sequence, that is N-terminal cleavable matrix targeting sequence and non-cleavable internal targeting sequence. N-terminal cleavable matrix targeting sequence, that is first type of MTS, N-terminal cleavable, which can be cleaved. N-terminal cleavable matrix targeting sequence are rich in positively charged basic amino acids such as arginine and lysine, and they also contain hydroxyl group containing amino acid. They are serine and threonine, but they lack negatively charged acidic residue. These signal sequence direct precursor protein to the mitochondrial matrix, inner mitochondrial membrane and intermembrane space. So this N-terminal cleavable site, if a protein containing this type of, I'll write it as N-terminal cleavable matrix targeting sequence. If a protein contains that, this can be directing the precursor protein to mitochondrial matrix, inner mitochondrial membrane and intermembrane space. To all these three locations, this protein can be directed with the help of this signal sequence. This sequence is cleaved once it reaches the matrix. It is removed by a peptidase called mitochondrial processing peptidase. That resides within the matrix. Consider we have the mitochondria here and the protein is trying to get inside. The protein is having N-terminal cleavable matrix targeting sequence. As there is a presence of this signal sequence, it can be directed, the protein can be directed to 3 matrix, inner mitochondrial membrane or intermembrane space. See, this is matrix, this is inner membrane, and the space between outer membrane and inner mitochondrial membrane is termed as intermembrane space. So the protein can get into this and this. After entering the protein which is having this N-terminal cleavable matrix, signal sequence or targeting sequence is broken down by the help of MPP, that is mitochondrial processing peptidase, which is residing within the matrix. And the second type that we discussed, the second type of mitochondrial targeting sequence is non-cleavable. As the name itself suggests, it is not cleaved. These are present in all outer mitochondrial membrane proteins. The proteins having non-cleavable internal targeting sequence is only targeted to the outer membrane. These are present in all outer mitochondrial membrane proteins and also intermembrane space and inner mitochondrial membrane proteins. In all intermembrane space, inner mitochondrial protein. So let us quickly get what we learned. So N-terminal cleavable matrix targeting sequence, pre-sequence, which are rich in positively charged basic amino acid. They have hydroxyl group containing amino acids. These signal sequence direct precursor proteins to the mitochondrial matrix, the inner mitochondrial membrane and intermembrane space. These sequences cleaved once it reaches the matrix by the help of mitochondrial processing peptidase. And it is the second type is non-cleavable internal targeting sequence. These are present in all mitochondrial membrane proteins, intermembrane space and inner mitochondrial membrane proteins. Next, we will learn about targeting of mitochondrial proteins into the mitochondria, especially to the matrix. Targeting to matrix, the protein which are to be targeted to the matrix will enter the mitochondrial matrix with the help of N-terminal cleavable matrix targeting sequence, which is termed as pre-sequence. These proteins, the proteins the proteins which are having N-terminal cleavable matrix targeting sequence gets into this mitochondria are synthesized by cytosolic ATS ribosomes and are maintained in the unfolded or loosely folded conformation to pass through the complexes and is carried out by cytosolic chaperone, that is HSC70. Let us now learn how this is done. So, to matrix, the protein is having N-terminal cleavable targeting sequence, right? So, there is a Tom complex. Tom means what? Translocase complex for or of outer membrane. So, the Tom complex, the Tom 20, especially the Tom 20 of Tom complex recognizes this pre-sequence, that is N-terminal cleavable matrix targeting sequence, and it transfers the precursor protein to central receptor, that is Tom 22. What does it do? First, Tom 20 of Tom complex, which is present in the outer membrane of the mitochondria, recognizes this N-terminal cleavable matrix targeting sequence. After recognizing it, it transfers the precursor protein to the central receptor Tom 22. This matrix targeting sequence, the MTS which is attached to that protein, it directs the protein through the Tom complex. Consider there is a complex like this. This MTS directs the protein through the Tom complex to the next complex, that is STIM. STIM complex is present in inner mitochondrial membrane. So, TIM23 complex remains associated with, now where we have come, so the protein of our interest has entered into that Tom, and then now it is entering into TIM complex. This TIM complex, that is especially TIM23, is associated with PAM. PAM means PAM. PAM means pre-sequence translocase associated motor. This PAM, what are we discussing? TIM23 complex is associated with PAM, that is known as pre-sequence translocase associated motor, which we call it as PAM in short. So, what all components are present in this PAM or this motor? This motor is having mitochondrial HSP70 co-chaperone, nucleotide exchange factor NEF, and other proteins. So, what all do they have? The PAM, that is pre-sequence translocase associated motor. This motor consists of mitochondrial HSP70 co-chaperone, nucleotide exchange factor, and other proteins. So, our protein with the help of the N-terminal cleavable matrix targeting protein has entered into TIM complex. What happens here, as PAM is associated with this TIM complex, the MTHSP is the component of PAM, right? This MTHSP70 binds to the translocating polypeptide chain, and drives its movement to the matrix. It drives the movement through the matrix in a reaction cycle of ATP hydrolysis. To drive that, the MTHSP70 drives that after binding to the translocating polypeptide chain, it drives its movement into the matrix with the help of hydrolysis of ATP. The pre-sequence of protein enters the matrix. The pre-sequence of the protein enters the matrix now. After entering into the matrix, there is a presence of MPP, that is mitochondrial processing peptidase. So the pre-sequence of protein enters the matrix, where the pre-sequence is removed. It is removed with the help of MPP. What is pre-sequence? That is nothing but N-terminal cleavable matrix targeting sequence. Many imported matrix proteins are passed on to another chaperone, that is HSP60. This HSP60 helps the unfolded polypeptide to complete folding of the polypeptide chain. Let us understand the process with the help of the flowchart and image. First, matrix targeting protein bind with the TOM complex. TOM20 of TOM complex recognizes pre-sequence, that is N-terminal cleavable matrix targeting sequence, and transfers the precursor protein to the central receptor TOM22. This matrix targeting sequence directs the protein through the TOM complex to the TIM23 complex. TIM23 complex is associated with PAM. What does this PAM have? PAM have mitochondrial HSP70, co-chaperone, nucleotide exchange factor, and other proteins. So MTHSP70 of PAM binds to the translocating polypeptide chain and drives its movement into the matrix in a reaction cycle that requires hydrolysis of ATP. Pre-sequence is removed with the help of mitochondrial processing peptidase when it comes to the matrix. Many imported matrix protein are passed on to another chaperone protein, that is mitochondrial HSP60, which folds the polypeptide chain. See, this is the image. So it is having signal sequence. Precursor protein is having signal sequence. It binds to the import receptor, that is TOM, the receptor protein in TOM complex, and insertion into the membrane by TOM complex inserted. Then it gets into the TIM23 complex, and it helps in translocation into the matrix by TIM23 complex. There is cleavage of the signal peptidase. It is cleaved and the proper folding is ensured. Mature mitochondrial protein will be get inside. So this is the complete image. So first one is HSP70 escorts polypeptide synthesized on cytoplasmic ribosome to mitochondria with the pre-sequence associated with TOM20 or 22. The basic pre-sequence leads the polypeptide through the translocase of the outer membrane, that is TOM, across the intermembrane space to the translocase of the inner mitochondrial membrane, that is TIM. The potential across the inner membrane pulls the pre-sequence through TIM into the matrix, where it is cleaved by the matrix protease MPP. The polypeptide binds matrix HSP70. The cycles of HSP70 binding to the peptide, followed by ATP hydrolysis and dissociation of HSP70 from TIM44 ratchet the translocating peptide into the matrix where it folds. So this was about targeting of mitochondrial protein into the matrix. So in our next class, we will be learning of the topic targeting of mitochondrial protein to the inner membrane of mitochondria. Thank you.